It is time to start this session of the SRS, and I'm going to start by introducing the first speaker. But before I do that, I want to let you all know that if you have questions of this session, put them in the chat to the host. I'll be the only one who sees those questions, and let's get going. So our first presenter is Christoph Potta, and the title of this presentation is "The Manganese Complex of the Rigidified 15-membered macrocycle, a comprehensive study."
Thank you so much for the introduction and welcome everyone. I'm just gonna start this. All right, so today I'm going to talk briefly about the project that I finished last year, the manganese complex of a rigidified 15-member macrocycle. So as we all know, metal complexes are frequently used in non-invasive diagnostic imaging. The most common is the magnetic resonance imaging, MRI, and they use gadolinium complexes in these measurements. Gadolinium-based complexes are used to enhance the contrast of the images generated based on the water content of the soft tissues. Here we can see a schematic representation of how those gadolinium complexes look like and how they interact with the water molecules. This is how we can get from a non-contrast enhanced image to a contrast enhanced image where we can detect abnormalities in the tissues, in the soft tissues, for example, in the brain. But the main problem with these gadolinium contrast agents is that free gadolinium ions are toxic. They can lead to nephrogenic systemic fibrosis, and a long-term gadolinium retention can lead to other side effects in the central nervous system. So how can we solve this problem, the problem of the toxic gadolinium ions? We can replace the metal center, for example, with manganese. It can lead to the manganese enhanced magnetic resonance imaging. If we compare these two metals, we can see that manganese is a little bit less effective in the contrast enhancing than gadolinium, but it lacks the toxicity of gadolinium at lower concentrations, and it's a calcium analog, so it allows the mapping of the central nervous system. So this is a structure of another manganese complex that has been approved by the FDA for clinical use, but has been discontinued because of commercial reasons. What issues do we have to solve with these manganese-based contrast agents? The main problem is the dissociation and the release a lot of these manganese ions into the extracellular space. So how can we solve this? There are proven strategies to overcome this adverse effect, and this is the rigidification of the backbone of the organic molecules encapsulating the metals ion. And they have, I'm showing two examples of this rigidification with different acyclic and macrocyclic ligands. So we try to apply this strategy for our molecule. I'm showing the synthetic route, how this molecule was synthesized in our lab, and then we made the manganese complex. On the left, you can see the solid state structure, the crystal structure of the ligand itself. On the right side, you can see the structure of the manganese complex with the manganese ion in the center of the cavity with two water molecules coordinated to the metal center. The next step was the characterization of this manganese complex. We found that it has a planar structure with two water molecules on each side of the planar structure. Due to the increased rigidity of the ligand of the organic scaffold, this leads to a little bit slightly lower complex stability, but it leads to a larger contrast enhancing potential, which is comparable to the FDA-approved gadolinium contrast agents. Another interesting side effect of this study was that we found that this manganese complex catalyzes the disproportionation of hydrogen peroxide, as we can see on this image on the right. We can see that our just manganese by itself does not catalyze the hydrogen peroxide disproportionation as good as our manganese complex does. And this shows that this complex can mimic the activity of the manganese catalase enzymes. And I would like to thank my advisor and my collaborators and everyone in the Green Research Group. And I would like to thank you for your attention. And I welcome any questions.
Thank you, Christoph. I don't see any questions in the chat, so I'll encourage people to put them in the chat. And actually, if you'd rather ask your questions verbally, I think we have that option too. I don't see any questions, so I'm since nobody else is asking, I'll ask one. So you're doing this research. I'm just kind of curious what your plans are for the future. Obviously, you're interested in this area.
So for the future, so this is a completely finished project. So we found that the slightly lower stability kind of prevents the use of this manganese complex.
Right.
You need different structures. You need a macrocyclic backbone, but with slightly different structures. It proved to be an unsuccessful manganese complex, but we had some interesting side effects as well, some other results.
Okay, thank you. Well, I think we have shy question askers here. So our next session will not start till 2:23. So, but I don't know that we need to keep you on the hook for no reason.
And stay here.
you stay here and watch. Well, just in case somebody has something to ask. Anybody else that wants to pipe in with a question, that's fine. The next one won't start till 12:23.
All right.
And you are a graduate student. You're working on a master's degree?
So this is one of my projects. My other project is synthesis of other macrocyclic ligands for the treatment of Alzheimer's and other neurodegenerative diseases. So it's a slightly different project, but the core is the same. It's the synthesis of those macrocyclic ligands and the characterization of their transition metal complexes.
Okay.
It's just the application is different.
Okay.
Oh, I found it. I did it.
It is now 12:23, so as we're ready to start our second presentation. And again, as I said before, if you have any questions, put them in the chat to the host, and as the host, I'll read questions out loud. Our next presenter is, I think, is Jacques Hernandez. You can correct my pronunciation if it's wrong. And the title is Searching for Twins of PTF11KX.
Thank you. And it's pronounced Jack, but it's fine. And also, thank you for that introduction. So yes, as they said, my name is Jack Hernandez, and I am presenting Searching for Twins of PTF11KX. And PTF11KX is just a name of a particular supernova. And here in the background of my first slide is an artistic rendition of a supernova. And what a supernova really is an exploding star. And these stars explode because their mass has become too great to be stable, leading to that explosion and leading to all that mass material, all that mass stuff being thrown back into the universe so it can later create even more stars later on down the line. And when these explosions occur, they make a ton of brightness, so bright that it can actually outshine an entire galaxy for a number of months. And since they're so bright and for so long, their light can actually be used for measuring tools and as measuring sticks for a lot of research. So if you ever heard anything about like the expansion rate of the universe, or like hot words like dark matter and dark energy, some of that research is used with supernovas. And as I said, PTF11KX, or I'll be calling it Target 1 for simplicity, is one of these supernovas. And it's particularly unique because it can stay bright for even longer times than what is seen in a typical one. So here in the plot right here is a typical supernova is shown in the orange, while our target 1 is shown in the green. And while between day 300, 400, a typical supernova will begin to die off. It's really hard to see anymore. It's starting to become basically embers. Our target 1 will still be significantly brighter for who knows how long. And the reason that is because there is a sort of like material blanketing around this explosion. So while the star is exploding, it's then hitting a whole bunch of other stuff, leading to even more brightness. And so far, there really hasn't been a supernova found to be like our target 1. So which is what my project is concerning. So we were liking to find more of these. And what we did is we used the Zwicky Transit Facility, ZTF, to compile a list of about 120 confirmed supernovae and to investigate them further to see if they are like our target 1. And we had all this data, and what we did is we compared the data before explosion and then after explosion, and subtracting them and taking a difference value of them called a flux. So here in the GIF right here is showing just one of these points of data before explosion compared to after explosion. And essentially we subtract them and got a number value. And so we would have multiple of these. And then we would have multiple of these and we would plot them over a number of days, obtaining a thing called a light curve, which is essentially just how does the brightness look like over a span of time. And out of this 120 list, we have 2 really points of interest, 2 supernovas that really popped out to us. So here in the plot is just comparing a typical supernova with our target 1. And in the blue dots is like out of that 120 list, how do they all on average look like? And on average, all of those tend to look like a very, very typical, what we expect to see. And in the orange and the green, those are our 2 points of interest. These 2 supernovas look very much like our target 1. And so these supernovas will require even more follow-ups to really delve in and learn more about them. So in more of a conclusion, we're able to find 2 more of these supernovas that were similar to our target 1, and it would require further investigation to like really be certain. And in any future work is to obtain light curves for even a larger supernova sample, so not just the 120, but maybe double, even triple that. And also using other surveys like the Legacy Survey of Space and Time, LSST, to widen that search as well. And that concludes my presentation.
Thank you very much. And Jack, I think I got it right that time, right?
Mhm.
I just decided I wanted to do some Dutch pronunciation or something. There's supposed to be people, if people have questions, they can put them in the chat and then I can read them off. But there aren't any questions. There weren't in the last presentation either. Maybe that doesn't work so well. It's all new agey, right? So if somebody else wants to even chime in with, verbally with a question, that's fine. But since I don't see any questions right now, I have a question for you. It's just about, I'm kind of curious how you do this kind of research in Fort Worth, Texas with all the lights. So I know you said something about getting data from the Zwicky Transient Facility.
Mhm.
What is that? And I'm kind of just, it's sort of a general question about astronomy research when you live in a city with bright lights.
Mhm. So the Zwicky Transit Facility, it's actually based in California. It's like they have a whole bunch of telescopes and they're able to take data like every day at a certain, not every day, every night, at a certain time of night, every day, and just like collect all that data. So it's all continuous and it's pretty dark over there as well. So there's no worry about like bright lights or anything. And this is all public access as well. So I just like hop on the website and download the data I need and be able to do research with it.
Oh, very cool. So is it in like the deserts of California? Do you know? I'm kind of curious where they find dark spots in the United States.
I believe it is. I know I don't have this like where it's at specifically in
okay.
California off the top of my head, unfortunately.
Oh, okay. No, I'll just Google it. It's fine. I just assume it's out there. You know, there's very deserty places in California where, you know, obviously it's not in LA. So it's somewhere out probably where nobody lives, I assume, but they have big deserts in California. So I'm guessing it's out there somewhere. Just curious. I don't know if anybody else has questions. It's kind of a funny format, and I don't know how well it works with the questions, but anyway, I do have one question, another question since I'm here. If anybody else is on and wants to ask a question, please interrupt me. I just have another question. You're an undergraduate, and so I'm sort of curious what your future plans are, either as research or graduate school or whatever. What you plan to do with your astronomy?
My goal so far has been like attending grad school after I graduate here from TCU.
Oh, okay. Is that this year or next year?
Next year. I'm a junior currently.
Oh, okay. Very cool. Do you have a grad school in mind, or?
I have a few grad schools in mind, but I haven't really like committed to one. There's like a couple in Michigan that I'm looking at, and also one in Washington that does research similar to what I did here. But I think so far my heart is very set on like investigating more about supernovas and just learning more about them.
Okay, thank you. I actually see somebody raised his hand, Peter French boy, and I don't know how to unmute him, but be fine. If you have a question, that'd be great.
Part of what I wanted to state is chat is disabled, so you can do it. Ask any questions through the chat. So that's why you're not getting any questions through the chat. So that's a note to this. To answer your questions to Jack, ZTF is at Mar Mountain in California. So.
Oh, okay. And how is the chat disabled? I see it, but then I guess it's disabled. Somehow you can only chat with me, I think.
Now I can. So I think that's fixed.
Is it better now?
Yeah, I can see it now.
Okay, so yeah, we need to change some settings, but now they can chat with the host.
Oh, cool. Okay. It was kind of blank, so. And I have 3:32, and we actually on the schedule from 3:31 to 3:35, at least the schedule I have, it just says 0. So I don't think anybody's on right this second. The next one starts at, looks like 2:39. Yeah.
Okay, then I will stop my share further now.
Thank you, Jack.
All right, thank you.
So I think everybody can just go on whatever they want to do for a little break here, and we'll reconvene and check your email, whatever you need to do. At 3:39, we'll go to our next presenter. I have a suggestion to go ahead and leave time. Is that from Lucas?
Yeah, it's me.
Oh, okay. I'm trying to figure, it just says dot.
Oh, that's right.
I think that's okay. I mean, if you guys think that's fine, is everybody here? As long as everybody's here, I don't mind moving ahead. I know.
Yeah, I'm ready to go if you.
Yeah, okay. No, that I'm absolutely fine with that. I know that my instructions will start exactly on time, but I don't mind breaking rules. So if Jack is ready to go, I'm ready to start. So and this would be Jack Joyce. So is you good? Okay,
Yes.
Okay, good. So here we go. I guess I'll do the introduction real quick. So Jack Joyce is our next speaker. Excuse me. Talking about assessing genetic diversity in northern yellow bats killed at wind energy facilities.
All right, I will go ahead and share my screen here. right. So my project is on assessing genetic diversity in northern yellow bats killed at wind energy facilities. So first, I'd like to thank the CERC for providing the funding that made this project possible, as well as to Dr. Hale and Dr. Williams for their support during this project. So wind energy is growing rapidly in the United States and has become the #1 source of clean renewable energy in the US. However, it's been linked to increasing bat fatalities, as you can see in these images here, through blunt force trauma and barotrauma. And this brings up concern for the persistence of bat populations due to their long lifespan and long reproduction rates. Interestingly, Texas is actually #1 in installed wind energy farms, but there's very little known about the anthropogenic impact that they have on bat populations. This is mostly due to the very few peer-reviewed publications coming out of the region. Bat conservation is of great human concern due to the fact that they make up about 1/5 of mammalian diversity and provide several ecological services in pollination and through the consumption of agricultural pests. The northern yellow bat, Lyserius intermedius.
Jack, I'm going to
Sorry.
interrupt you just for a second.
Sure.
For whatever reason, I cannot see your presentation, so I'm not sure why that
really?
is. It's just,
Okay.
I can hear you, but I can't see the.
It's all
presentation.
black, but you are co-host, so you should be able to share your presentation.
Okay.
Try stop sharing
says
it during
I'm
the game.
screen sharing, but I'll try to stop it and restart it.
Yeah, because we can see you properly in your spotlight. So you're
Okay. Can you see it now?
no.
Hmm.
So let me share the presentation for you.
Sure.
One second. Okay. Can you guys see this?
I can see it now.
Okay.
Okay.
So if you want to scroll down to the next slide here.
Should I maybe decrease the?
Yeah, if you want to zoom out, maybe. There we go. That's good. Okay. So I'll just kind of re-summarize what I've said here. Essentially, wind energy is growing rapidly in the US. It's becoming the #1 source of clean renewable energy, but it is linked to increasing bat fatalities, as you can see in these images here, through blunt force trauma and barotrauma. And so this brings up concern about the persistence of bat populations, and their conservation is of great human concern. So on the next slide, we'll start talking about specific things to northern yellow bats, also known as Lyserius intermedius. There are 2 subspecies, the Lyserius intermedius floridanus and the Lyserius intermedius intermedius. This map shows their range throughout the eastern United States, the Gulf Coast down to South Texas, as well as parts of Mexico and Central America. And interestingly, southern yellow bats, Lyserius aga, will also share parts of this range in South Texas. And so it's possible that some of my samples will be a mix of northern yellow bats and southern yellow bats, which will provide some interesting data as to which species is most adversely affected. And then on the next slide, we'll talk about some previous studies that have been done in collaboration with Texas State University, where mitochondrial DNA sequencing and microsatellite genotyping characterize the genetic diversity and estimated effective population sizes for Lyserius aga and Lyserius intermedius species at Hidalgo and Starr counties. Specifically at Starr County, about 8,000 bats were killed per year for all species, and the northern yellow bat was found to be the second most affected species. The current project on this next slide is to expand on this existing research in the Rio Grande Valley region of South Texas. So I have 18 wing tissue samples that were collected from Willacy County in 2015, and this population has actually never been studied before. So I'll amplify and sequence the cytochrome C oxidase 1 locus of the mitochondrial genome and compare these results to Hidalgo and Starr counties. DNA was extracted following ammonium acetate isopropanol precipitation method and amplified by PCR at the 550 base pair region of the cytochrome C oxidase 1 gene in the mitochondria, sequenced using ABI big dye terminator cycle sequencing and analyzed on an ABI 3130 genetic analyzer, which is pictured here. These sequences will be compared to GenBake voucher sequences in order to obtain a species ID. And this is where I'm able to tell whether the sample that I'm working with is from a northern yellow bat, a southern yellow bat, and even which subspecies of northern yellow bat I'm working with. And finally, unique sequence haplotypes were detected using a program called GenALEX. For anticipated results, this table comes from a former grad student, Austin Chips, from the Hidalgo and Starr counties. And you'll see in that column labeled N, that shows the number of carcasses collected for each species of bat at these wind energy facilities. And then the capital H represents the haplotypes or unique DNA sequences present in the population. The lowercase h represents the haplotype diversity, which is a measure of the percent difference in haplotypes present in the population. And then pi is the nucleotide diversity, which is a measure of the percent difference in single nucleotide polymorphisms present in the population. So of great interest is this haplotype diversity measure. And essentially what these numbers mean is for the Lyserius aga, the southern yellow bats, for example, if you were to take 2 bats from this population, there's essentially about a 2% chance that they would have unique haplotypes. In comparison to the Floridanus and intermediate subspecies, that's closer to a 60% chance or a 54% chance. So the southern yellow bats are experiencing much less population diversity in comparison to the northern yellow bats. And the results that I obtained from Willacy County will be compared to these results. Into the discussion, simply more knowledge is needed about population structure and diversity to assess the extent to which bat populations are being impacted by wind energy facilities. And my data will have the potential to fill this knowledge gap. Specifically, this is going to provide a baseline population diversity for this population that's never been studied before, which will allow scientists to return to these sites in 5 to 10 year intervals to reassess diversity. If we find that there's been a drastic decrease in population diversity, this is probably serves as pretty good evidence that the populations are being adversely affected. Furthermore, we need to talk about jurisdictional issues that prevent informed decision-making in regards to wind energy development. Typically, bat fatalities are not a concern that are addressed in this decision-making process. And furthermore, the U.S. Fish and Wildlife Service provides only voluntary guidelines for wind energy producers to avoid impacts to wildlife from wind energy. And so there's really no laws in place to protect these bats. And this shows that continued mitigation studies need to be emphasized to improve predictability of bat fatality. And this paves the way for collaboration amongst Texas scientists, Texas Parks and Wildlife Department, and wind energy companies to continue wind energy development in a responsible way that conserves these threatened species. So that can finishes off my presentation unless we have any questions.
Thank you, Jack. We do have a question from Dr. Richardson, and I think this is okay. His question was, do you have a hypothesis on whether the wind turbines impact the bat significantly?
So I do believe that it is significant. And now from that one wind energy facility in Starr County, we saw that 8,000 species or 8,000 bats are killed per year for all species. This is only one wind energy facility in a rather significantly developed region of the United States for wind energy. And across the United States, it seems to be that these bat populations are producing or are experiencing a lot of deaths due to wind turbines. I believe in North America, it's hundreds of thousands per year. Now, the issue with that is that these are cryptic species. They travel alone. They are tree roosting. We don't know a lot about their life cycles and their behavior. So estimating their exact population sizes is really difficult. What that means is that we don't really know what percentage of the population is being killed. If there are millions of bats in these populations, maybe that's not as significant. But if there's less than that, it could be quite significant. And I do believe that they are, but it is very challenging to estimate that.
I don't, anybody else that has questions, it would be great to hear. Have some more, you could put them in the chat and I'll read them out. I'm not seeing one just now. I'll ask my favorite question. So I know you're an undergraduate. I'm just kind of curious. Are you going to continue the research, or I don't know what year you're in, or maybe you're a senior.
Yeah, I'm a senior, so I'm actually going to dental school next year.
Oh, okay. Well, I don't know what that has to do with bats, but but congratulations.
Yeah, not a lot of dental specific research at TCU, but my interests in science are pretty diverse. So I'm happy to be learning about anything I can. So.
Most excellent. Where are you going to graduate? Where are you going to dental school?
Iowa College of Dentistry.
Okay. All right. A little cooler up there.
Yeah.
So any other questions from the audience? I think we're good, and thank you. Wait, actually, I think we got one. It just showed up. It said, well, actually, you have to work on the. Oh, actually, it's a joke. It said you have to work on the back teeth, so it wasn't a question.
Yeah.
It was a comment. It's all good. Thank you.
Thank you.
And we have a couple of minutes for the next presentation, and it's been pointed out to me by Bob Bell, and he's absolutely right that we probably should start on time because there's going to be some faculty that might be coming at exactly that time to see the students and maybe even friends and family. So we'll stick to the schedule exactly, even though we're slightly ahead. So we got about two more minutes for the next one. It is 12:47, so it's well, in my time, 2:47. And I want to introduce our next speaker. It's Rose Betty Hernandez, and please correct my mispronunciations. I didn't get that right. But the title of the presentation is Mapping Landslide Susceptibility and Ground Displacement Assessment of Travis County, Texas, Texas, USA.
Hello, you can hear me?
Yes,
Okay, you can see my screen?
yes, we can see it.
Okay, awesome, thank you. Okay, good afternoon, everyone. Thank you for being here, connecting and listening to my first poster presentation about my research topic, which is the mapping landslide susceptibility and ground displacement assessment of Travis County, Texas. So a little bit about my presentation is that the landslides can occur naturally or are triggered by human activity and can cause massive damage and financial cost. In this study, we will deal with the analysis to identify areas susceptible to creep, being a slow scan of landslide. Once that the susceptible areas are identified, analysis for ground displacement is applied using the synthetic aperture radar interferometry technique. For RTK, GPS measurement and geology soil sample for analysis. So the area of my study is the area under study is Travis County, located in South Central Texas, and the area has experienced rapid growth of population in the past few decades, and development has incursion into unstable terrain that is vulnerable to landslides. So this is the map of the spatial location of my study area, and this is a landslide that occurred in the past May 2018. So the major of my presentation of my study is identify and analyze areas susceptible to creep, quantify the rate of displacement occurring during landslide, and also identify the factors that trigger the hazards. So that data and method, the methods that I applied was a geological map, land cover, DEM of 30 meters of resolution, Sentinel-1 SAR image. And my methodology was I applied GIS technique, empirical approaching to get the landslide susceptibility index, and also with the integration of the landslide susceptibility index and the IMSAR SPAS, it will lead to identify a slow movement ground displacement that will be validated with the work field using the RTK GPS and measurement and the geology and soil sample for analysis. as a result and discussion of my research is that landslide values classify into three susceptibility categories, how we can see here in my map on the right. So we have the slow susceptible. The lowest susceptible is the blue, moderate is yellow, and the high susceptible is the red one. So, and also we can see that the Shoal Creek landslide that affect this area is located in the high susceptible areas. So also the highest factor that trigger the hazard are the greater degree of a slope and unconsolidated material with clay content around the urban areas. Our analysis showed a good spatial correlation with the Shoal Creek landslide, how I mentioned before, and the interprogram analysis, this is the interprograms show a strong displacement anomaly in the Shoal Creek area. So this is we are talking about here the Shoal Creek area, and we can see that is an anomaly showing here. So efforts are currently underway to quantify the displacement over 5-year intervals using the SRS technique. As a conclusion of my preliminary study is that the EIS approach in this research is to assess potential slope instability in Travis County. This analysis delineates susceptible areas to landslide by 3 levels, low, medium, and high, based on preliminary triggering factor as the degree of the slope, rock salt composition, and land uses vegetation as characteristics. Preliminary interferogram results validate that Shoal Creek area had ground displacement that can be validated in the further analysis with SRS technique and RTK GPS measurement in the field. And finally, the identification of areas susceptible to soil creep will be useful for effective landslide management that can support the decision maker for urban and infrastructure plans. So that's it. It's my presentation of my poster.
Thank you very much. We have a question coming from Dr. Richardson, and his question is, which of the different kinds of slope movement are the most prevalent in the Travis County area, and do they match specific colors on the maps? And he also adds, in parenthetically, I assume the colors are the slopes. So that was kind of a question.
Yeah, the colors in this map is the division between low susceptible to landslide, medium susceptible, and high susceptible. So the kind of landslide that we are trying to identify is the slowest one, that is the creep, because they can be predictable. So we don't have because Travis County is pretty much plain. It has a low slope. So the ones that they are affecting in this area are pretty much the creep landslide.
And I'm not seeing other questions. Does anybody else have any questions they want to put in the chat? I see Kathy Calvert has raised her hand. Do you want to speak out? Or Katie?
Hi, thank you. I was wondering why you were trying to detect creep landslide and not another kind of landslide.
Yes, because the creep landslide can be predictable with this technique. So we are using data from 5 years. So data from 5 years from satellite data, sorry. So the change on something, we can detect the change of if something's happening from in only a slow movement. We're talking about millimeter to centimeter from 1 year. So we are able to detect this movement from just creep and not the other ones.
Well, thank you so much. I think it's about time for our next presentation to come on. So thank you very much for your presentation. So we'll start the next one in about a minute. It is time for our next presentation. This will be by Madison Braley. And the title of the presentation is Family Trees of Black Holes. And if you have any questions, just shoot them in the chat to me, the host, and I will read them out at the end of the presentation. Not seeing her coming on yet.
Hi, I think you let the wrong person share their screen. I can't share mine.
I just did it. You have the same name.
Okay, thank you. Okay. Hi, everybody. I'm excited to get started talking y'all about my research on the first stars titled Family Trees of Black Holes. So during the first 100 million years.
This is the host. Oh, there you are. Never mind. I didn't see you, Madison. You weren't spotlighted. I can see you now.
Thank you. So during the first 100 million years or so after the Big Bang, the universe was dark and there weren't any stars yet, but the Big Bang created hydrogen and helium, which were the ingredients for the first stars that we call population 3 stars. So hydrogen and helium are lighter elements themselves, and their spinning motions don't cool their stars as efficiently as heavier elements do in stars today. And as a result, we think that population 3 stars ended up getting extremely massive, sometimes over 1,000 times the mass of our own sun. And to visualize that, you can think about one of the dots on the screen as the mass of the sun and all of them as the hypothetical mass of just one population 3 star. So the big question we're trying to answer is what exactly was that mass distribution among the first stars? So the easiest way to figure that out would just be to look at them, but we can't actually look at them with current or upcoming telescopes. So instead, we have to look at signatures of population 3 stars to try to get an idea of what they looked like. We use simulations based on some educated parameters to help us hypothesize the possible function of the mass distribution of the population 3 stars that we call the initial mass function. Now, keeping in mind that these stars are really massive, when they get to about 100 times the mass of the sun, they can collapse directly into a black hole as seen in the diagram to the right. And these direct collapse black holes were so massive that they were able to merge throughout time into supermassive black holes that are littered around the universe today. And based on observations of these supermassive black holes, we can trace up their merger trees, which are like family trees, to simulate the first direct collapse black holes that came from the first stars. So remembering that our goal is to identify the initial mass function, in other words, the distribution of masses of the population 3 stars, we expect this function to be top heavy, meaning that a greater number of these generation of stars will be on the more massive end of the scale. So first, we compare predicted slopes of the initial mass function, and this is shown in the top plot where we compare our simulated mass functions to a theoretical function that's shown in gray. And then for 4 different slopes of the predicted initial mass function, we compare the number of black holes per mass. And to do this, we identify simulated masses of population 3 stars where black holes have formed. And the population 3 stars randomly populate the function based on parameters like the size of the dark matter halo that they're in and how much gas is available to form stars in the first place. And this simulation resulted in what you see on the bottom of the screen, which is what we would expect, that the simulated population 3 stars have a top-heavy initial mass function for all of the 4 functions that we ran. And once we've confirmed that the mass function for the simulation is what we expected for one run of each potential slope, we run a simulation for each slope individually where the halo gets randomly populated at least 100 times. And we want to look for a convergence of the most black holes produced for a given slope of the initial mass function. We want to find the function with the most outputs because it should include all the direct collapse black holes that have formed for that halo. And these plots on the right of the screen are also heat maps. So the lighter colored hexagons indicate that more runs outputted in that number of black holes for that specific mass. So you can think of them as kind of an overlay of all the runs that we've done. And right now we see the most convergence around a slope of 0.5 for 100 runs, but we have to increase the number of runs to achieve minimal uncertainty, as the law of large numbers tells us that the more that we randomly repopulate these simulations, the more accurate of an initial mass function we're going to be able to get. With all of that said, our simulation tells us that the initial mass function is top heavy, and we understand that the slope of the initial mass function will become more clear with more runs of the simulation. So next, we want to find an upper mass limit for the direct collapse black holes that we run to find out if there's an upper mass limit for the population 3 stars in our simulation. And the combination of discovering the slope of the initial mass function and defining an upper mass limit will lead us to hopefully answering our question of how massive were the first stars. So thank you all for listening, and I'm happy to answer any questions.
So you have the option of putting questions into the chat and I'll read them out. Another option is if you want to raise your hand, you can just ask it out loud. Looks like we have a quiet group this time. Maybe you're relieved anyway. Does anybody have questions? I'll just ask a question about, going forward. You're an undergraduate, so are you? I don't know. I don't on my sheet. I don't see junior or senior, so I was curious if you're going to continue this research.
I am a senior, but I'll be continuing this research through the summer since I get to stay an extra semester.
Oh, okay, excellent. So I think we're fine.
I have a question. I can read a question in the chat right here. Somebody's asking, why is it super important that we study the first stars?
Well, the first stars kind of created the background upon which modern stars formed. So the first stars form without any what we call metals. So that's basically anything heavier than helium. And we see stars today are super enriched with metals, and so much so that we can be alive today. Like we have more than just hydrogen and helium in our bodies. So we want to basically figure out the chain of events that happened from the birth of the first stars all the way up to today to figure out how that happened.
And I have another one, which I think at this point is from your dad, is asking, are black holes related to supernovas?
Sort of. So supernovas are, let me go back to one slide here. Supernovas form in, can y'all see my cursor? This section here, and also in this middle section here. So it's 2 different death options for stars depending on how massive they ended up getting to be.
I think we're about due for the next presentation. So in the interest of being on time, I'll just thank you for that, Madam.
Y'all.
Who's our next presenter?
The next presenter is going to be Richard Vanderbroek. One up right now. Is he there?
Yes, he is.
Okay. And I'm not seeing him yet. Oh, I see. He's coming on. Richard, are you coming on?
Hello, can you all hear me?
Oh, I can now.
Oh, how's it going? I'm sorry. It's giving me an error message saying the host stopped the video.
No, you're allowed now to share because you needed to be co-host and I was waiting for you to be ready.
Okay. So how's it going? Give me a second.
That's okay, Richard. I'm the actual host of the whole this whole session. So I'll introduce you while you're getting ready.
Sure.
Let me make this disappear a little bit. So this is Richard Vanderbroek, and his presentation will be on particle motion in a rotary dryer. You can, at the end of if you have questions, put them in the chat and I can read them out at the end. I wasn't seeing the questions during the last session, so just in case I don't see your questions, feel free to raise your hand and ask your questions out loud too. You can get called on. So we're all ready to go.
And one quick question before I go. I also have 2 other people here. Are they going to be allowed to present or no?
Who are the 2 other people?
Alexandra Skopek and Katie Erickson.
Yeah, do they need to share their screen
No, I will be in charge of the screen. They're just gonna go ahead and speak.
and talk? Yeah, so let me, Alexandra, and who else?
Katie Erickson.
Okay, Alexandra is already co-host. Katie Erickson is co-host now, so they can all unmute themselves and speak.
Perfect. And can y'all see me?
No, we can't.
I cannot right now.
I don't know why it's not giving me the option to show my face. I apologize. Is it fine? Can I just go ahead and start?
Yeah, if you
Okay.
share your screen.
Is it not sharing?
No.
Oh, it says it's screen sharing. Can y'all see it now?
I just see your blank name. I
Yeah.
see your screen now.
Okay,
Yeah.
we good? All right, sorry, this computer's being a little wonky. We good? All right. So how's everybody going? My name is Richard Vanderbrook, and I'm doing the WAST Rotary Dryer project. And our sub-team for this project was tasked with modeling the particle motion inside of a rotary dryer. So as previously stated, our senior design section was tasked with designing and modeling a lab scale rotary dryer. And this machine is able to mine dried limestone by lifting and displacing material that is fed through a hopper system, which you will later see in this presentation. It is able to accomplish this drying process by using what we call lifters that are attached to the inside of the cylinder. So as the cylinder rotates, the pieces of metal that are attached to the inside will lift it up and push the material through in a certain pattern. So our team was tasked to model how the particles of limestone will fall off as the drum itself is supposed to rotate. And so keeping this in mind, we kept certain parameters vital to creating these systems of equations, such as the lifter type, lifter length, the dryer rate of rotation, and the incline of the dryer as well. So our approach. When we were studying this falling motion, we assumed there was no friction, there's no centripetal force present, and no bouncing of particles as they're being transported through the actual cylinder itself. In kind of keeping this in mind, our team used basic kinematic equations along with simple particle modeling to create a system of equations that will govern the particle's trajectory as it is to be thrown off the lifter. And from this, we're able to provide what we call a curtain or a plot of the particle's X and Y position as it falls off of the lifter.
So here is a model that we created using Inventor. And as you can see in there, there is the cylindrical dryer that's mounted on its base. And then it's also connected to a feed system, which is being designed by a different subgroup, but it provides the purpose of transporting the product from the exit back to the entrance of the cylinder. And this video is showing this the dryer in motion with product in it. So the first lifter type that we modeled was called the radial lifter. And the reason that we began with it was because it has the simplest geometry. So you can slightly tell from this picture, but it's 4 inches long and it extends inward from the edge of the cylinder. And we took the approach of modeling a particle starting at the very edge of the cylinder and determined the curtain of product coming off the lifter at various angular positions within the cylinder as it's rotating. And so the picture on the left is the dryer in motion with product moving through it. And the picture on the right is the plot that we created with our model. And you can see how they compare with each other.
So next we moved on to a 90-degree lifter. This is different than the previous because it has an extra leg on it, creating that 90-degree angle. Our model and calculations found that the material would ideally begin to fall in the second quadrant and would completely empty on that side. And this is due to the geometry of the lifter and cylinder combination. There's a photo of the dryer in motion on the left and our Excel plot on the right, which can be compared to verify our model. Finally, we set out to find the residence time in the dryer. This is the total time it takes for the limestone to enter and exit on the other side. The calculation was based off the angle of the cylinder along with how fast the cylinder was rotating. We found that approximately 71 seconds was the time it took for the limestone to enter and exit the dryer. Thank you so much for listening to our presentation.
Thank you to all three of you. Any of the members of the audience that have questions that they would like me to read out, if you put them in the chat, I can read them out. Another option is if you prefer to raise your hand and ask the question yourself, that's also fine. And you're on, Katie, so you might be the question answer. I'm not seeing questions. I have my own questions. So I understood the presentation. I'm kind of curious exactly where these type of dryers work and where they appear in industry, what they're used for. So where they appear in the industry is that when limestone is mined, it comes out of the ground with a lot of moisture content in it. Okay. And so we are building a lab scale model of one that's used in the industry. And in the industry, it's a huge rotating dryer to where the wet limestone is put in on one side. And as it rotates, hot air is blown through and the limestone is dried on the other side, which will then be used and transported for purchase and sale. And when you say huge, how huge? Just out of curiosity. I'm not sure off the top of my head, but I want to say maybe maybe a 4-meter diameter. Okay, thank you. And I have one really quick question. I know that we have another presentation coming up right now, but the one question that I did see in the chat is, where do you want to take the model from here? If you can give a quick answer. We would like to make our model very user-friendly so that our client will be able to enter in any inputs that they want, RPM, angle, and it'll spit out the exact path of the limestone. Okay, thank you very much. Appreciate it. Thank you.
Thank you.
And our next presentation is going to be loaded up in just a second, and this will be Gregory Tomlinson, and he will be speaking about evaluating class reorganization in equivalence-based instruction.
Hello, can you hear me?
Yes, we can hear you.
Perfect. And can you see my screen?
Yes.
Yep.
Beautiful. Okay. Hello, everyone. My name is Gregory. Today, I'm going to be talking to you a little bit about equivalence-based instruction and stimulus equivalence. So stimulus equivalence is essentially just how we treat physically dissimilar stimuli as equivalent to one another. And the example that I always use is 4. So the number 4 and then the written FOUR and 4 dots are all equivalent. They look very different, but we treat them all as the exact same. And so the research that I'm looking at is, what is the best way to teach these relationships and establish these stimulus classes? Is it on the left here, EBI, which is equivalence-based instruction, or is it on the right, which is complete instruction? So on the left is equivalence-based instruction, and you see a lot less arrows, and so this assumes there are a lot less directly taught relations. So if we teach that A goes with B, A goes with C, A goes with D, we hope that they assume that B goes with C, and then C goes with D, and so on. So it leaves some up to interpretation into the participant. Whereas on the right here, we have complete instruction. And this is our control condition in which every single one of the relations is directly taught. So now I'm going to be talking to you a little bit about our procedure in this specific study. And so first, we started off with an ABCD training. And like I said, we have 2 groups, the EBI group and the CI group. OTM, it just means one to many. So as you see in the EBI group, it's one to many. And so there are a lot less trials. There's only A, B, A, C, and A, D, A, B, C, D. Whereas in the CI group, you see a lot more number of trials. And so we wanted them to reach mastery criterion in order to see that they actually established these classes. So mastery criterion, they needed to perform 89% correct or better in order to qualify for the ABCD test. And this is what we actually analyzed. So they took a test, and these are the results that we see. In that, we saw that the accuracy actually didn't differ between groups. However, the EBI group took far less trials in order to get to the same accuracy as the CI group. So it actually may be a little more efficient. And this validates our findings in previous studies. We've been looking at EBI research for a few years now. But now what's different about this specific study is class reorganization. So reorganization is essentially just what happens when the rules change. What happens when A1 no longer goes with B1, A1 goes with B2, A2 goes with B3, and so on. So we entered all the participants, both groups, into the same reorganization training. And the reorganization training is a little more similar to EBI. So I'm going to talk a little bit about why that's important later. But we changed the AB relations. So AB no longer goes together like they thought, but the AC and AD relations remain the same. And then we entered them into a test, just like we did in the ABCD. We tested them on it to see how they did. And actually, our results were really interesting. Our results do show that there's a difference in accuracy and number of trials. The EBI group performed better on the test, and they reached that with less trials than the CEI group. And this is really, really interesting to us. And I talked just a second ago about how class reorganization is an EBI group. And so this is important because the CI group initially, of course, the ABCD training, they did CI training, which is all of these trials. But now, once they're thrown into class reorganization training, they have a far fewer number of trials. And so this is more EBI, whereas the EBI group, they were thrown from normal EBI training right back into EBI training. So we're wondering if they were just used to the method of EBI and teaching, so they were more receptive to that. And we're actually looking at that in a study that we're conducting currently. But what these results really show us is that the equivalence classes produced through EBI were far more flexible than the classes that were produced through complete instruction, and that they were able to change more easily when they learned new information. So I believe that's all I have to say, but I'm welcome to answer any questions if anyone has any.
Thank you, Gregory. Anybody that has questions, feel free to put them in the chat and I'll read them out. Or you also could raise your hand and just ask them out loud yourself. Perhaps people are wearing out as the afternoon goes on. I'll ask a quick question. You're an undergraduate, so I don't know if you're a junior or senior. I didn't know that you were planning on sending this research further.
Yeah, I'm a junior actually, and I've been in the lab since my first semester sophomore year. So I've actually been looking at EBI research since then, and it's been really exciting. But this is a study that we did last year, and we're actually currently doing a research, as I said, like a little follow-up to it, in that we're just changing the class reorganization so that it matches more of a complete instruction versus the EBI that we see here. So we're just changing a little bit about the class reorganization. And so we just want to see if these results stay the same.
Okay, well, thank you very much. I appreciate we all appreciate your presentation, and thank you. And I think it's about time for us to move on to the next speaker.
Thank you.
Thank you.
Hi, everyone. I'm gonna share my screen.
And we'll wait to start just one more minute. And the only reason, Maria, is because just in case somebody really wants to see this and they think it starts in 1 minute. In fact, what I'll do is I'll just kind of I'll go ahead and introduce you, and then we'll wait just until the, well, you know what it is 3:19 now, so we can start. So this is Maria Amoras, I think.
Yes.
Okay, and she'll be speaking about SMO volunteering system.
All right. Hi, everyone. I hope everyone's doing well today. I am Maria Moros. I'm a computer science major here at TCU. And today I'm going to talk to you alongside with Lydia Papi about our senior design project, which is the design of the TCU and UNT volunteer scheduler. So I think Lydia can unmute, but I can present this part. So our project was to design the volunteer scheduler system, as I was saying, of the TCU and UNT Medical School. So students in the School of Medicine are required to complete certain hours of volunteer opportunities and in various nonprofit organizations around Fort Worth. So currently our client, Dr. Bonnell, has to keep track of all the volunteer opportunities and student volunteer hours by hand and in Excel, and has to communicate with students about the opportunities over email. The system we have developed is meant to make this process much easier for everyone.
I had a question in the chat. Somebody is wondering if you're sharing your poster. I know that I'm seeing it in a very, very small font, so I don't know if you could make it a little bit bigger.
Yes, I am sharing.
I don't think that that's your presentation. We are seeing your email.
Oh, my email. And can you see what the web page here?
What we seem to be seeing is we can't read your email, but it's because the font's too small. But what we're seeing, yeah, you're right. It looks like the email as opposed to your presentation.
If you have any problem sharing your presentation, I can share it for you. But try again.
I wanted to demo here. Let me close all of this. Can you see now the website?
Yeah, TCU and UNT volunteer scheduler.
Yes.
Yeah, that's what we can see.
All right. So I wanted to show you a little bit here our website here. So the idea is to make everything easier for the students and for the Dr. Bonnell, which is the service learning director, to create events and for students to sign up for events. So here we can see the calendar, which students can see and sign up for events that they're interested in. Also, they can check in and check out for those events, check the month, the year, the time, and check in, and also check out. And and all of these will be saved in our database. And also the administrator will be able to create reports of those hours that were completed by the student here by putting their first name, last name, and also we'll be able to create reports based on event ID on what is going to be saved in our database. Also, the medical school staff will be able to see and manage events right here. Dr. Bonnell will be able to see all of the events, view the events, the attendance, who has signed up, who is in the wait list, and also modify those events as well as cancel them in case the event is not going to occur anymore. And that's about it. If I show you here my script, my poster, are you able to see it?
Yeah.
Yes.
All right. Yeah. So here are some of the technologies that we use for the backend. Maybe some people are not very familiar with it. The backend is basically what the user doesn't see. It's the database, it's the data flow. And we use an SQL database, which is a relational database to save our data of the students and the faculty. And the front end is what the user interacts with, which is what I just showed you. And we use a framework called Vue.js and as well as HTML and TypeScript to create that user interface. And also this semester, everyone from the team learned an amazing new technology, which is Firebase, which is a Google Cloud service. And Firebase is helping us with our user management and also our authentication and our passwords and all of that. So probably you have all seen whenever you log into some pages, they give you the option to log in with Gmail or with Facebook and all of that. And that is because Firebase is doing the authentication for that website. And that's what we're currently doing in ours. Let me know if any of you have any questions.
And for questions from the audience, you have the option of putting your question into the chat. I'll see them as host and I can read them out, or you can raise your hand and speak out that way. I'm not seeing any questions. I'll just ask, are you planning on extending, you're an undergraduate, I know, so are you planning on extending the project further?
It's pretty much done. And we are already testing it, so the medical school can start using it right away.
Okay.
Mm-hmm.
And here is a question. It says, how many volunteer hours does a medical student, I think, do per semester? I'm not.
For now is we believe it's 75. That's what we have in our program. But the administrator can change that anytime in case the requirement changes. So he has a place where he can change that, and it will require those specific hours for the students.
Okay, thank you. Are there any other questions from the audience? I don't hear any. Thank you very much, Maria. Enjoyed your presentation.
Thank you.
And reading your email. We really couldn't read it. It's fine. Our next presentation will start in 2 minutes. We'll wait till the time.
This is a little interesting. I can see our next presenter 5 times on the chat. So I don't know which one is the actual one to make co-host. So I put them all as co-host.
Okay.
Sorry, that's me. I think whenever I sent the link to my family, it made them
Okay,
as...
so probably all of the members of your family are co-host now.
Okay.
But Anne-Marie, don't let him talk. So we'll start in just a second.
Okay.
Just want to wait till 27 after, in case more members of your family want to show up.
Can I go ahead and share my screen of my...
Yeah, absolutely. In fact, it is time now. So let me introduce you. This is Anne-Marie DeMarco, and she will be speaking on blood alcohol concentration, open field activity, and sorry, it's on my screen. I just lost it. And the CFOS expression after consumption. So you're on stage.
Well, first of all, thank you everyone for joining my session. My name is Anna Marie DeMarco, and I'm an undergraduate research assistant in Dr. Papini's lab. So this project that I'm going to present to you was focusing on the behavioral and neurological effects of high alcohol concentration in rats. So we were interested in investigating this topic because we wanted to understand the underlying mechanisms for the rewarding properties of alcohol, especially at high concentrations, and why people are likely to seek it out. So from previous research, we know that high alcohol consumption can alter many areas of the brain, including the dopamine output and the nucleus accumbens, which is basically the pleasurable feeling when you do something rewarding, like getting a good grade on an exam. So we see the same effect in alcohol's presence. So many previous studies looking at alcohol's effects have used rats as models for consumption, but this is sometimes difficult because rats find high alcohol concentrations aversive. But we found that our rats here at TCU in a pilot study will work to receive high concentrations of alcohol up to 66%. So we wanted to continue to explore this phenomenon and evaluate the specific consumption in a paradigm and its effects on open field activity, whether it changes blood alcohol content and the activity in certain brain areas connected to reward. So to test this, if you look at the timeline now, we wanted to measure their alcohol consumption versus water during separate sessions. So rats were split into 2 groups based on whether they received alcohol or water first to counterbalance the order. Then they were put into several different consumption trials. So for the consumption trials, the rats would remain in their home cages, but they differed on whether their bottle was filled with water or alcohol. So to control for spillage, the rats had yoked controls that had a bottle containing the same liquid, but did not have access to the sipper. These trials lasted 1 hour. So following the 2nd and 4th trials, the rats were put into an open field. So this open field is a large box where they can move around freely, and there's a camera mounted on the ceiling that records their activity so that we can evaluate their locomotion. So 10 minutes after the open field, the blood was drawn from the tail vein to calculate their blood alcohol content. So our study found that the rats consumed about the same amount of water and alcohol when given access in a 1 bottle consumption test. They did not reject the alcohol. So as you can see, we found significantly high levels of blood alcohol content, which indicated that they did consume the alcohol. And it's right here on the open field, alcohol and water. And so there was a positive correlation between higher blood alcohol content and the more the rats would move around, especially in the periphery. So after the 5th and final consumption trial, the animals were perfused and their brains were extracted so that we could measure the neural activity in the various brain regions. So to do this, we stained for a specific protein called CFOS, which is expressed in recently depolarized neurons, which makes it a really good indicator of overall activity. So basically, the more CFOS dots that we could find in the stains meant the more neural activity. So we measured multiple regions, but we focused on the insular cortex and the anterior cingulate, where we found more expression in females that had consumed alcohol compared to those that consumed water. There was also higher CFOS counts in the nucleus accumbens shell. As you can see, it's a little hard to see, but so this one right here is the alcohol. So you can see the darker dots in the core and in the shell compared to the rats that had water. And so the insular cortex is specifically involved in the integration of body systems and has been linked to addiction, in particular alcohol use disorders. And then the nucleus accumbens and the anterior cingulate cortex are both involved in reward processing. So overall, we found that the rats did not prefer nor reject the high concentrations of alcohol, and that this consumption significantly raised their blood alcohol content, which suggests that they did consume it and it promoted locomotion in open field. So together we can interpret these results as a form of sensation-seeking behaviors in which they find alcohol an interesting substance that they're willing to interact with. Future studies may investigate the effects of high alcohol concentration on other learning paradigms and the complex interaction between alcohol, the brain, and behavior.
Thank you so much, Anne-Marie. Anybody in the audience that would like to ask questions, you can do that by putting them in the chat and I'll read them out. Another possibility is you can raise your hand and everybody's welcome to ask questions. I don't see any. I've noticed that the questions have kind of gone down as the afternoons. It's getting later in the afternoon. I have a quick question. Just in terms of future research. So you're an undergraduate,
yes.
Either junior, senior, I'm not sure, but you're planning on expanding, doing sort of extending the study?
I'm not necessarily planning on doing that because I am a graduating senior, but I know that,
fair enough.
yeah, but I know that our lab is wanting the next steps for this kind of experiment. Would be interesting to change things like the environmental conditions. And so our rats are socially isolated, food deprived, and they're in wire cages. And so we've collaborated with a lab in Spain that's seen different effects. And so just kind of changing that, those kind of environmental conditions we think could change, as well as changing the alcohol concentration.
How did you end up collaborating with the lab in Spain, just out of curiosity?
Dr. Papini actually collaborates with, I think it's University of Seville.
Oh, okay. Okay, so the advisor. So does anybody else have questions? Well, thank you so much. And it's Anne-Marie, isn't it? I think I've been saying Anne-Marie. And just an announcement to, because this is the last one in our session, that the award ceremony will start at 4 p.m. Using the link on srs.tcu.edu. So if you would like to see that, it's coming up relatively soon.